In the previous video, we discussed about gametogenesis. Gametogenesis is the process which by a diploid or a haploid precursor cell undergoes cell division and differentiation in order to form new gametes. Right? We can remember that. But this process had two steps. The first one was the common pathway and the second one was the individual pathway. Why the first step was known as the common pathway? Because in males as well as in females, the pathway seemed to be very similar. What happens? First thing, we gave a name to the precursor cells. We named those precursor cells as primordial germ cells, right? These primordial germ cells migrated to the gonadal ridge. How did they migrated? They from the dorsal endoderm migrated through the hind gut. Fine. Once these primordial germ cells reached the gonads, they were known as gametogonia, and the singular term of gametogonia is gametogonia. From there onwards starts the individual pathway. In the females are a different pathway, and in males is a different pathway. Now in this video we are going to focus in the male's pathway, and it is known as the spermatogenesis. I on YouTube, I am Antru Sinthujan. Let's directly jump into spermatogenesis, and there is one more additional information. This gametogonia in males were known as spermatogonia. Fine. But there is another thing. Once it reached the gonadal ridge, it was known as A1 spermatogonia. This is small in size, and the nucleus had egg shape. Fine. In general, spermatogenesis has three phases. The first phase is known as the spermatocytogenesis, where the spermatogonia becomes spermatocytes. The second phase is spermatidogenesis. These spermatocytes become spermatids. The third phase, spermiogenesis. These spermatids become the sperm. Now we are going to look at how it produces the spermatocytes. Okay, this A1 spermatogonia becomes the A2 spermatogonia, and then this A2 spermatogonia becomes the A3 spermatogonia, and this A3 spermatogonia becomes the A4 spermatogonia. There's a speciality. The whole process takes place under mitosis. Mitosis is a cell cycle. We will discuss it in a different video. So mitosis takes place, so remember that. And this A4 spermatogonia has three options. What are the options? The first option, it can self-renew. It has the ability to self-renew. Not just A4, all the spermatogonias has the ability to self-renew. Same thing, A4 has this ability to self-renew as well. Or else it can undergo cell death, which is known as apoptosis. We will be discussing about apoptosis in a different video. But now just remember apoptosis. The third step, what is important for us, it becomes as an intermediate spermatogonia, and this intermediate spermatogonia is becoming type B spermatogonia. Type B spermatogonia is important. Why? Because it is the last cell to undergo mitosis. So once this type B spermatogonia starts mitosis and become the primary spermatocyte, the second phase starts. This primary spermatocytes is the last place where we can find it is as a diploid cell, and it starts to undergo meiosis, and it is a large cell with a large nucleus. Now the first egg shaped nucleus has become larger. These primary spermatocytes become the secondary spermatocytes. Once the primary spermatocyte undergoes the meiosis, and it becomes a haploid cell. But the thing is, here only the nucleus can be seen as a haploid nucleus, but it functions as a diploid cell. But then again, it undergoes the secondary meiosis, then becomes the spermatids. Once it becomes the spermatids, it undergoes the third process, where it is the maturation process, which is known as spermiogenesis. And again, spermiogenesis has 4 steps. What are the 4 steps? First one is the Golgi phase. Second one is the cap or acrosome phase. Third one is the formation of the tail. Fourth one is the maturation phase. What happens in the Golgi phase? Golgi phase, now in every cell we have the Golgi apparatus, right? In Golgi phase, what happens is the Golgi apparatus starts to produce lysosomes. These lysosomes are known as acrosomes, fine? And these acrosomes form and travel in to one side of the cell. Okay, in the second step, the cap phase, what happens is these acrosomes form as a cap around the nucleus, and then on the opposite side, the mitochondria collects. All the mitochondria gather on the opposite side. When on the third phase, formation of the tail, what happens is the centrioles of the cell starts to generate a tail known as the flagellum with the structure of 9+2. Okay, that's how the sperm starts to get the shape. The final phase, the maturation phase, what happens is, it's very complicated. The thing is, the maturation happens like at the last minute, and we know a cell has cytoplasm, right? In the maturation phase, a little bit of cytoplasm will be absorbed by the adjacent cells to this sperm cell, and those cells are known as Sertoli cells. These Sertoli cells absorb some of the cytoplasm, and they produce some fluid. So that's why the sperm get the mortality. Once it matures, sperm is a mature sperm. So that's how one part is produced. But just a sperm is not enough. We have to talk about ovum as well, oogenesis. We will be discussing about oogenesis in a different video. Until then, see you soon. Bye bye. I'm Antru, Sinthujan.